In order to understand how the muscle contracts, first let's identify the structures of the muscle. This structure is a fascicle or bundle of muscle cells. Each fascicle is usually made up of 10 to 100 muscle fibers and muscle cells. Here you can see just 4 of the muscle fibers. Surrounding each fascicle is a paramecium, which is connective tissue that surrounds and protects the muscle fibers. Around each muscle fiber is a cell membrane called the sarcolemma. Here you can see it in blue around each of the muscle fibers. This has special functions that allows the muscle action potential to travel through it. Thousands of tunnels of the sarcolemma run from the surface of the cell towards the center of each muscle cell. These are called transverse tubules or T-tubules, and here you can see them how they go through the muscles. The action potential travels through the sarcolemma and T-tubules so it can quickly spread to all of the muscle fiber. Also located in each muscle fiber is mitochondrion, shown here in yellow, which produces ATP during muscle contraction. The mitochondria is strategically close to the muscle proteins that use ATP. Each muscle fiber is filled with tiny threads called myofibrils. The myofibrils extend the whole length of the muscle. Surrounding each myofibril is a sarcoplasmic reticulum, seen here in yellow. The sarcoplasmic reticulum is a membrane sac filled with fluid and calcium. You can see here the blue dots are our calcium. There's 10,000 times higher concentration of calcium in the sarcoplasmic reticulum than outside of it. This is possible because the sarcoplasmic reticulum also has calciquestrin molecules, which are shown here in purple. The calciquestrin is a calcium-binding protein that allows the calcium to be sequestered and stored at this high concentration level. Now we are going to show how an action potential occurs in a motor neuron. An action potential which travels down the axon causes calcium ion channels to open. The calcium is indicated by these red ball-like structures that flow through the calcium channels into the presynaptic terminal. When these calcium ions flow into the presynaptic terminal, it causes these synaptic vesicles, indicated by the dark green circles, to release neurotransmitters. These neurotransmitters are called acetylcholine and are represented by the light green and pink squares. These acetylcholines are released from the synaptic knob by exocytosis into the synaptic cleft. The synaptic cleft is the open space between the motor neuron and the muscle fiber. Once the acetylcholine is diffused into the synaptic cleft, it attaches itself to an acetylcholine receptor, which is indicated by the blue-like structures. Once 2 acetylcholines are attached to the 2 ends of the acetylcholine receptors, as shown here, the receptors open up. Once the receptor opens up, it causes sodium ions to flow from the synaptic cleft to the muscle cell. This is shown by the green ball-like ions. At the same time that this is occurring, potassium ions, indicated by the black ball-like structures that are present in the muscle cell, move along an open acetylcholine receptor into the synaptic cleft. An action potential is generated and is indicated by the black zigzag line that flows across the muscle cell membrane. Acetylcholine is then rapidly broken down into acetic acid and choline by an enzyme called acetylcholineesterinase. The yellow-like structure in the synaptic cleft indicates the acetylcholineesterinase. Here, it shows the breakdown of acetylcholine into acetic acid and choline. Once this occurs, the choline is then reabsorbed by the presynaptic terminal and combined with the acetic acid to form more acetylcholine, which enters the synaptic vessel. This process will then start over. An action potential is a wave of voltage that can travel on certain cell membranes. This occurs when a neuron sends information down an axon, which is always away from the cell body. This sends the pulse or wave of electrical activity that is created by depolarization. The depolarization allows a positive current to flow inward. A stimulus causes the resting potential to move towards 0 millivolts. When the depolarization reaches the negative 55 millivolts, or the threshold, a neuron then fires an action potential. After this stage, there is the repolarization phase. This is where the positive charge causes a reversal of the membrane potential to a negative inside and back to the resting potential. The resting state of the cell membrane is negative 70 millivolts. This is the sarcomere, which is made up of the thin and thick filaments of a muscle fiber. Thick filaments are myosin, represented in red, and thin filaments are actin, represented as pearls. Z-band is the end of each sarcomere, represented as an orange zigzag. The A-band is the dark zone, with the overlapping of thin and thick filaments. The H-zone is where the thick filaments are located. And the I-band is the lighter region where thin filaments are located. That was good. That was better. Tropomyosin lies on the 2 sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. When calcium ions bind to the troponin, as indicated with the blue circles, troponin changes conformation and moves the tropomyosin. The black dots on the pearls are the myosin binding sites. This uncovering of myosin requires binding of ATP. After ATP binds, it is split into ADP and an inorganic phosphate. Energy is utilized to prepare the power stroke. In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.